Now we'll discuss the ground tissues. Okay, so inside the ground tissue system, you know that there are parenchyma, collenchyma, and sclerenchyma. Okay, they are made up of all these three type of cells. Now these are the simple cells. Okay, now consist of all tissues except epidermis and vascular bundle. So they don't contain a vascular bundle because when you see this ground tissue, this is the pink colored structure here. They don't have a vascular bundles. Together with that, they are simple tissues and not the complex tissue like xylem and phloem. So this contain parenchyma, collenchyma, and sclerenchyma. So we have studied this. Now you can see all these three cells are present here. Parenchyma cells are present in cortex, pericycle, pith, and medullary rays in the primary stem and roots. So when we discuss the stem and roots, so where it is present, it is generally present in the cortex region. Okay. Together with that, it is present in the pericycle. What are this region? We'll see. These are the pericycle and cortex region. In the pith, also present, and medullary rays. So this medullary rays helps in the transport of the water in the radial manner. So in leaves, the ground tissue consists of a thin walled chloroplast containing cell and it called mesophyll. As parenchyma are also present. So in the leaves also it is present in a thin walled structure chloroplast containing cells and it calls mesophyll. So the cell name is mesophyll which also contains the chloroplast. So you can see here spongy mesophyll is present in the leaves. Now vascular tissue system, so it consists of complex tissue. Okay, so this is the ground tissue system that we have studied. Now we'll study the vascular tissue system. So it contains a complex tissue like xylem and phloem. We already learned this. So xylem and phloem, they are made up of multiple cells and they together forms the vascular bundle. It is present in the center. You can say in the center it is present. So it also have a primary phloem and primary xylem and secondary phloem and secondary xylem. Now, xylem phloem, based on the presence or absence of the cambium, vascular bundles are two types. Okay. Now, as you can see, where cambium is present between the xylem and the phloem. So, if it is present, then it's a there are two types: open type and closed type. So, we'll see that. What are this? So, this is the epidermis, the outer layer. In the cortex region, ground tissues are present as we discussed. So, here xylem and phloem are present and the cambium is present in between them. So you can see here open type the cambium is present. Okay, so open type when the cambium is present and closed type when it's absent. So vascular bundles can form secondary xylem and phloem tissues. When the cambium is present, then it also forms a xylem and phloem secondary tissues. And examples are dicotyledons. And in case of monocotyledons like maize, cambium is absent and they do not form secondary tissues. You can see here open vascular bundle cambium is present and closed vascular bundle cambium is not present between the xylem and the phloem. Now vascular tissue system, based on the arrangement of xylem and phloem, there is two type. One is radial. Okay, so if it is a radial, it helps in the transportation of generally water and the food in the radial manner, means the symmetrical manner. Now xylem and phloem are arranged in an alternate manner on a different radii. So you can see this is the radial one. Xylem and phloem, they are arranged in alternate manner. So arrangement is like this, then it's a radial. If arrangement is not like this, then it's a conjoint. So conjoint can be open and closed. Like it contains a cambium, then it's open. If it doesn't contain a cambium, then it's a closed. Xylem and phloems are joints situated at the same radius of vascular bundle. So they are joined like this in case of conjoint, seen in the generally in the stems and the leaves. Now we'll see the dicot and the monocot plant. So dicotyledons root. First we'll see the root. Okay, inside the root what is there. So when you see the TS, transverse section of the root, you will find that this part is nothing but the epidermis. It outer part, the skin color part, root hair and epidermis. In between cortex is there, and in cortex parenchyma, collenchyma and sclerenchyma cells are present. And in the center xylem and phloem is present. Okay, phloem is present, xylem is present, some protoxylem, metaxylem, these are nothing but the primary and the secondary one. Meta is the secondary one and primary is the proto. So in case this metaxylem, the secondary one is present in the center and this proto is present at the periphery, and it is opposite in case of shoot system. So it contains epidermis, cortex, endodermis and stele. So in case of epidermis, it's the outer layer, cortex where the ground tissues are present, endodermis means the inner part, and then stele is present. So this is the outer layer. Okay, many unicellular root hairs are present. Then in the cortex, parenchymatous cell is present, which are thin walled and made up of lignin. Then after that, in endodermis, it's the innermost single layer of the cortex. You can see innermost single layer of the cortex. Barrel-shaped cells without intercellular. So this is new one, endodermis. You can see this is the barrel-shaped cells. Okay. The tangential and radial walls of endodermal cells have a deposition of suberin. Now, when you see this endodermal cell wall, okay, inside that some depositions are there of organic compounds which are suberin, water impermeable waxy material. Okay, it is nothing but the organic compound, and it forms the Casparian strips. So it generally forms the Casparian strips. Okay, you can see here these are the Casparian strips. So arrangement is like that, and this is the radial wall. Now the stele part, tissues on the inner side of endodermis, they include pericycle. So when it is in endodermis, it's a xylem and phloem, and together with that it also contains a pericycle, a few layer of a thick wall parenchymatous cell which is inside the stele part. Okay, so after the endodermis, the whole part is stele. Now initiation of lateral roots and vascular cambium during so here all the vascular things are present, vascular tissues are present, they are nothing but xylem and phloem. Pith is innermost region, it is the small and inconspicuous. You can see pith, this is the innermost region. Now vascular conjunctive tissues are present, for example parenchymatous cell lying between xylem and phloem. So these are conjunctive tissues. Why? Because they join both the cortex and the endodermis and the stele part together with the help of this parenchymatous cell which lies between, which present between the xylem and the phloem. And the vascular bundles are present because these are vascular tissues. So xylem and phloem patches are present, and also cambiums are developed. So then we'll see the monocotyledonous. So it includes epidermis, cortex, endodermis. Similar structure is there. What is the difference here? Pit is large, okay, and together with that sometimes cambium is absent, okay, in case of monocotyledons. So they also contain the same thing. They are usually more than 6 polyarch xylem bundles. So they generally contain more amount of xylem bundle, and here pit is also a large, and no any secondary growth. Secondary growth is not there. Now anatomy of dicot and monocot. So we'll see stem now. Okay, we have seen the root part. Now we'll see the stem part here.